Good evening, everyone, and a warm welcome to all attendees to our second session of the Nomic series. I'm Aarti Darba, bioinformatician working at Cartinos Healthcare, and I'll be your host for tonight's webinar and will also be leading the Q&A session after our formal presentation. Today, we have the esteemed Dr. Santosh Kuriakose, MS, a senior faculty member in the Gynecologic Oncology Division of the Department of Obstetrics and Gynecology at Government Medical College, Kozhikode, Kerala. Our webinar title, Does Ethnicity Influence Oncologic Outcome? Will delve into Dr. Kuriakose's research on the prognostic significance of the polymutations in endometrial cancer. Before we dive into Dr. Santosh's insightful presentation, I'll take this moment to acknowledge his remarkable accomplishments in the field of oncology. Dr. Santosh is the winner of the prestigious 2020 John J. Sciarra International Journal of Gynecology and Obstetrics Paper Award for his groundbreaking article titled Diagnostic Accuracy of Self-Collected Vaginal Samples for HPV DNA Detection in Women from South India, published in 2020. He is also a visionary leader who established the first gynecology oncology division in Kerala at Government Medical College, Kozhikode, in 2012. He has also been the recipient of the esteemed FIGO International Gynecology Oncology Fellowship at the British Columbia Cancer Agency, Vancouver, Canada, in 2015. He also participated in the esteemed UICC Technical Fellowship at Memorial Sloan Kettering Cancer Center, New York, US, in 2018. Without further ado, please join me in extending warm welcome to Dr. Santosh Kuriakose. The stage is now yours, Doctor.
Hello, good evening. The question of whether ethnicity has got anything to do with the oncology outcome came as a result of our research project, which we undertook at our institution of Government Medical College, Kozhikode. It is based on the unique mutation called Pol mutations in endometrial cancer. I'm a gynae oncologist and I work in my parent institution. The whole research was initiated by the Cancer Genome Atlas group, and they identified the genotypes of endometrial cancer in 2013. And this article that appeared in Nature 2018, and which shows this unique four types of endometrial cancer, which were determined genotypically, and they were having different prognosis. Biologically, they were different. And
Hello?
the most important or most unique feature of this classification is that they identified a mutation called Pol mutation, or which they classified as ultra mutated, because the huge number of mutations in that particular subtype. But unlike what was expected, they found that none of these patients actually recovered or died, which was quite unique in these patients. So we also wanted to know whether our patients also have this subtype. So that was there in our mind. A lot of investigators started investigating this, and they also independently verified this finding of the good prognosis of Pol mutation and bad prognosis of P53 mutations. Okay, now what we are interested is now in this Pol mutation subtype, and the three publications that appeared back to back from the Vancouver group, that is British Columbia group, Jessica McAlpine and Telhook. So they have got a promise discovery group, then promise validation group, and promise confirmation group. So all this has actually more than 600 patients, and they confirmed Pol mutated, all from Canada. Another group from Netherlands, that is by Ellen Stelu and Jalingbos, they also came up with the same analysis. And another group from US, led by Robert Soslow from Memorial Sloan. They also, but this is what I would like to, the key points that they brought forth in the discussion of this paper is that the Pol mutations are unlikely to be, they are so good that they will not progress to stage 3. And they even suggested that staging could be avoided in these advanced stage patients. because sequencing will is not required because they are unlikely. All mutated patients will not be seen in stage 3 patients. And they suggested that treatment like chemo or radiation, which were commonly given in grade 3 tumors, may not be required in these Paul mutated patients. So very intriguing finding. High risk as far as the pathology is concerned, but purely because of the presence of Paul mutations, they are having good prognosis. And sure enough, in 2021, the European Society of Oncology came up with this new classification where they classified even Paul mutated patients up to stage 2, irrespective of the pathologic types, whether low grade, high grade, LVSI, that they classified them as good prognostic group. So that is very, very non-conventional. Now, our research team aimed to do a TCGA surrogate typing of endometrial cancer patients, 150 patients we studied. The study was quite long, I should say 7 years, and we had 100 patients from my parent institution, GMC, Government Medical College, Kozhikode, and our nearby institution, private sector, MVR Cancer Center, both in Kerala. So we sequenced exon 9 and 13 of this polymerase epsilon gene, and the research was entirely funded by Science and Engineering Research Board funding and of around 25 lakhs. And the whole project was undertaken as multidisciplinary research unit, MRU, at GMC Calicut. Our findings were published in International Journal of Gynecology and Obstetrics, which is a phase journal of FIGO. Now, we titled it as High Prevalence of a unique Paul mutated patients, which has got a poor prognosis. This title itself gives the reason why that question arises. They are having a poor prognosis. I will dwell in detail about it. So we had 151 patients enrolled. We identified 39 unique Paul mutated patients and 25%. That is 25%. And the exon 9 and 13, as I already said, we sequenced these two mutations because we selected these two because 95% of mutations, polymerase epsilon mutations, are seen in these particular exons. And we could find 35, majority of them found in exon 9 and 6 of them in 13 and 1 in both. And we had both 9 and 13. Now, significant association was seen with Paul mutated patients having high-grade tumors, deep myometrial invasion, lymphovascular space invasion, and non-significant lymph node involvement. That's why I didn't put it there. We had quite reasonably good follow-up of 40 months. And to our great surprise, I should say, this polymerase epsilon gene carrying endometrial cancers now turned to a red subgroup. That is, they are having poor prognosis. This had a disease-specific survival of 51 months, which is 24 months less than that for Pol wild type, which is 2 years, which is quite significant. And the hazard ratio was 3 times hazard for having a disease recurrence or death due to this disease. So we were totally perplexed. I should say we were devastated. We never expected this. And of the 39 patients, 28 occurred in stage 1, and advanced stage, we classified more than one as stage advanced, and we found 11 patients in advanced stage. And we were even more devastated. This advanced stage tumors, which again carrying Pol mutated tumors, when we compared with advanced stage tumors not having Pol mutations, this Pol mutated advanced stage had a very dismal prognosis, which means to say they lived only 2 years, probably 2 years. So hazard ratio is 9 times more than the same stage, but they are not having Pol mutations. So that That is very surprising. But if we looked at the stage 1 tumor alone, they were having good prognosis. In the sense, they were not different from each other. Those having mutations in the pole or those not having mutations in the pole, they were not different. Now, this is what we call the 11 pathogenic pole mutations that the published literature has. So these, almost all the publications, the TCGA, the Vancouver group, the Netherlands group, all of them had these mutations. And they were what we call ultra mutated, having more than 100 mutations per megabyte. So basically, this ultra mutated group. Now, when we looked at our patients' mutations in the pole gene, we couldn't find a single mutation in that were listed in the previous slide. These were unique mutations, or what Jessica McAlpine's meta-analysis has reported as non-pathogenic mutations, within inverted commas, non-pathogenic mutations. And we did find the same mutations in the same loci. We found some mutations, but I will come to it in my subsequent slides. The mutation locus were common with certain studies that were conducted in China, and a lone study that was conducted in India that was from CMC Vellore. So we could identify our mutations to be quite similar to this Chinese and the only other Indian study. But the glaring disparity from the studies conducted in the West was that our mutations were not matching with their mutations. So that is something very interesting. Now, I will just take you through what this pole gene is. It is actually the polymerase epsilon gene, exonuclease domain, that is 200 amino acid residues from 271 to 85. They are highly conserved across species, and they are part of what you call the exonuclease function that actually is related to the proofreading function of the DNA. And then this is crucial for the DNA, its exonuclease function of the DNA, and that safeguards the DNA integrity over lifetime. Now, we could identify 34 of these unique and recurrent mutations in this particular segment. So it is very difficult for us to logically think that these mutations are not significant. These mutations should be significant. Looking at the various functions that it is doing. So in our study, we classified them as recurrent if they were seen in two samples or two patients, or in our samples or in other studies. And also, at least there should be it should be classified as pathogenic by at least 3 in silico models. Now, I already said there are 11 of the 28 patients, 11 pole mutated patients occurred in advanced stage. Now, there are 10 patients who have had a recurrence, and all except one all died. And looking at this patients who had a bad outcome, 7 of them are actually belonging to advanced stage tumors. Or in fact, 75 to 80% of our advanced stage patients died, who carried pole mutations died. So I already showed you the previous graph that the pole mutated advanced stage tumors are very high likelihood of death due to some uniquely probably because of this pole mutations. Now, we know that these advanced stage tumors will have definitely got some radiotherapy or chemotherapy as a part of treatment. And we wanted to minimize the effect of this radiation treatment or chemotherapy treatment. We use a mathematical model, what you call Cox proportional hazard ratio. And we found this that in a Paul mutated had a significantly higher disease-specific survival, disease significantly poor disease-specific survival, with a hazard ratio again 8.67 compared to Paul wild type. And there are two studies that has come up, which is quite early, published quite early, that it tells us that these Paul mutated tumors have got reduced sensitivity to commonly used chemotherapeutic drugs like cisplatin, paclitaxel, and radiation. So, and some increased sensitivity to less used chemotherapeutic agents, less cytarabine. Now, my first thought was that this could be because these patients actually had a bad outcome because of inherent inactivity of these tumoricidal activity, poor tumoricidal activity of these agents. Now, let us discuss about our findings. Now, all mutations were, as far as the studies were concerned, were non-pathogenic. And we looked at the published articles, and we found one study which appeared in a good journal. It was actually from the Shanghai First Mother and Infant Hospital, that is in China. And they found somewhat similar results to what we got. Out of the 400 plus patients they studied with endometrial cancer, 35 Paul mutations they identified, and 32 of them were non-pathogenic. And 3 were belonging to the allied with the TCGA subtype. And another thing, they aligned one more aspect. Those patients who got chemo and radiation had a poor prognosis. Another study from China mainland, that is, they found a subgroup of Paul mutated endometrial cancer patients with what you call MELF pattern, that is histological pattern of some certain inclusion bodies and something. So that particular pattern, patients having that particular pattern and Paul mutation, they have poor prognosis. Again, it's not a marginal elevation, 15 times higher hazard ratio. So this is another unique study which is from our own country, and this was done at CMC Vellore. They studied 48 patients, and they found unlike our study, they did Sanger sequencing from 9 exon Paul exon 9, 10, 11, 12 up to 14, all of them, and they identified 5 Paul mutations, and of this, 3 occurred as Paul mutation alone, and they had a good prognosis. Orange line has a good prognosis. 2 of them had both Paul polymerase gene mutation as well as p53 mutations. So 53, as you all know, is bad prognosis. So that subtype has got the worst prognosis. So this is that subtype. So one thing you should understand is our study and their study also allied in that they also got the same non-pathogenic mutations. They also, in their study, none of these 11 pathogenic mutations surfaced. And the 2 patients that died actually recurred and died where they were having this D440G and P4. That was actually personal communication from Dr. Rega Pai, molecular pathologist from CMC Vellore. And one thing I would like to grab your attention is this graph actually is different from the P53 graph. This is the green line. There is an argument that pops out that this poor prognosis of Paul and P53 is because of the presence of P53, which I would actually say a big no to that, because if had it been so, they would have carried they would have paralleled the same survival pattern of this P53 mutated subtype. So this group, which carries this Paul and P53, is a unique group of endometrial cancer. So that is the meta-analysis that was published, the only meta-analysis with respect to Pol mutations that were published by Jessica McAlpine from the Vancouver, British Columbia Cancer. And then she found that hazard ratio for this non-pathogenic Pol subtype was high, just as what we got, 3 times hazard ratio. And in their study, 18% were non-pathogenic. So you must understand that this meta-analysis actually have patients from United States, Canada, and Europe, except for one study from Singapore. So almost 95% of patients are from the West. So they don't have even at the West, they could find 18% of non-pathogenic group. So what do we have now? The whole question is, we have an Indian ethnic endometrial cancer patients who are having a subgroup of polymerase epsilon mutations that are unique in the sense that are not that much reported in the West. And they were having poor prognosis. It's very important that clinicians become more aware of this category. Otherwise, Pol mutated good prognosis. That is what is now in the clinical arena, especially among oncologists. So the clinicians has to be particularly be aware that it is not like that. It's not like that. And salvageability of our patients were very low. They invariably died and progress. And unlike what the Western researchers thought, they progressed to advanced stages. 1/3 of them present, so more than 25% presented in advanced stages. And especially in advanced stages, they had 9 times poorer outcomes than the corresponding advanced stages without Pol mutations. That doesn't mean that they don't have other mutations like P53 or something, some catenin B or beta catenin or something like that. There are other mutations. But in spite of all those things, this particular group has got bad prognosis. We don't know why. So that is there is the question that I have formulated for this this talk. Is ethnicity a determinant of oncologic outcome? So there are a lot of treatment implications from this research, whether we need to seriously think of using other chemotherapeutic agents in these patients. As you might be knowing, this Pol mutated has got very high mutation burden, and it invites a lot of peritumoral lymphocytes. And these peritumoral lymphocytes are the guardian angels, or that actually will fight the tumor itself. So the body itself fights the tumor. So but now, because of the effect of chemotherapy and radiation, this lymphocytic angels actually are destroyed. That is what we thought from the whole thing. And especially in advanced stages, this very protective lymphocytic infiltrates are actually, in fact, destroyed by this chemotherapeutic agents, which actually, in fact, doesn't have much effect on the tumor itself. So it is a double-edged sword in the sense. Or is it something related to the unique Pol mutations? Actually, are we dealing with a unique set of mutations that are having a bad prognosis? Or is it the ethnic influence per se? I discussed the findings with Dr. Jalingbos, he's from Netherlands group, and they also share the same concern as I am discussing with you, whether this is something unique. They also do not have an explanation why we didn't find a single mutation that is classified as pathogenic in the West. And very important is that FIGO has now reclassified endometrial cancer. FIGO, as you might know, is the International Federation of Gynecology, who put forth the staging of endometrial cancer. They have classified the stage 1, stage 2, high grade, low grade, myometrial invasion, with invasion, no invasion, with LBSA, low invasive, LBSA. If they have Pol mutations, they're classified as 1AM, that M stands for molecular subtype, and they are having good prognosis. Pol mutated have a good prognosis. That means to say that they may go even without any treatment apart from surgery. So just like any research, our research also actually brings out more questions than answers. And are there actually these fold mutations the primary event? What you call the key mutations that are causing this cancer, or is it driver mutation, or is it secondary event? We don't know the answer. In any case, I personally believe that these mutations, since they're occurring in this very significant exonuclease domain, highly conserved exo motif one, I think they are significant. They should be having significant biological effects. And the question I don't know is the answer I don't know is, do Indians really have a low prevalence of this pathogenic fold mutations? And I would be very interested to know if Indians having this 11 pathogenic mutations, do they have good prognosis as what is reported, but was actually reported from the West? My sincere thanks to my patients, whom even with all the diseases and their adjuvant treatment, they cooperated with us, and at Government Medical Kozhikode and Americans, and their families who gave their wholehearted support to us, and my co-investigators at our institutions, Adana Suraj, Shammi, Shaini, Amrita, Bindu, Naina, Nation sir, Sridharan sir, and Kavita Uma. I end my talk. Only the question is, the rhetoric question is, what is the truth? You will know the truth, and truth should be actually liberating you. So the question is, what is the truth? So thank you very much. So that's it.
Thank you, Doctor, for this insightful presentation on fold mutations. I will now welcome people to post their questions in chat so that we can take it up. So as they are putting it in chats, let me ask you few. I have few questions to ask.
Okay.
You mentioned that abundant in the exon 9 and 15.
Yeah.
And all these are characterized as non-pathogenic.
Yeah.
Won't you think that the mutations other than present in exon 9 and 15 could be pathogenic, and they could be contributing to the poor prognosis?
Yes, the answer is yes. To your question, the answer is yes. But the possibility of finding is, I should say, less than 10%, because 95% of those identified were identified in these 9 and 30. So the possibility is there, but it is less. You may identify a few if you could do the sequencing from, but the only other study that was done in India from CMC Vellore, they have sequenced from 9 to 14, and they couldn't identify a single mutation of this belonging to this 11.
Another question that I have is, when you showed that the stage 1 patients with fold mutated or wild type have the same survival rate, while the one stage they differ.
Yeah.
Would you like to comment on that?
That is very, I should say, I don't know why it is like that. I simply don't know, because advanced stage tumors carrying fold, we have actually given our speculations on that. The thing is, the point is, we are having unique mutations. And these mutations are having a different biological behavior. That is what my take on it. The second point is, these peritumoral lymphocytes are actually actually kills the tumor because of the effect of chemotherapeutics like cisplatin. These lymphocytes are actually coming down, and actually this tumor is resistant to the chemotherapeutic agents that we are currently using. So that's why I said it should be a double-edged sword. The protectors that what we give in the form of chemotherapy or radiation, in fact, actually destroy the good lymphocytes or what you call the peritumor lymphocytes. At the same time, they don't kill the tumor. So in fact, you are actually allowing the tumor to grow, or you are actually giving the tumor a good environment for it to grow actually.
Right. I have two questions in my chat. So first is, was tumor mutation burden assayed in these tumors?
I didn't get the question.
Did we
Can
check
you
the
repeat?
TMB levels? Yeah. So the question is, did we check the TMB levels in these samples?
No. We actually have done a Sanger sequencing. We have not done a next generation sequencing. So we don't know the mutation burden of these patients. So that is one thing very important you should understand. We do not know how many of them are ultra mutated. That we will know if you have another study or some other study where you do next generation sequencing and look at the mutation burden. That only that will clarify. So my answer, we have not looked into it.
Okay. And the second question is, were all pole mutated tumors were MMR fusion?
That is, we are still looking into the data. We have not completed with our data. We are not completed analyzing that data. We will come up with that. MMR. We have done the
And
MMR part and
P53.
P53. We have also done a few other mutations in this cohort, but we are now in a stage we cannot comment on that.
Okay. Let me know if there are other questions, and if people want to come online to ask.
Hello, Aarti. There are a
Yes.
few more questions towards Dr. Santosh from LinkedIn Live. One of the questions is
Okay.
by Deepshika Satish. The question is, can we use European data as a baseline data for formulating a database regarding nutrigenomics-based test for Indian population?
Very interesting question. We have a few researchers from Netherlands who are actually discussing the same question to us, whether they want to generalize their findings to the global population. So they also want to know whether their findings, by what we call their findings, in sense, predominantly their patients from the West, that findings actually can be translated as such to our patients. The point is, we have very little studies at the genomic level from India, very little. So just looking at the meta-analysis, you might know that. So generalize. So coming to the answer, if you ask me whether this data from the West can be generalized to to Asia or African countries, I will say that will be taking too much of a liberty, because we need our own data, and unless you have our data, you'll have to go by what they say. So basically, you need more research. So unless we need to have our data, so unless we had this cohort, and unless we had in sequence, we would just have to say that pole mutated are having good prognosis, and let us just not treat them anymore. Let us just observe them, and these patients would have died, may have had a even bad prognosis. So we need to have our data. That is the only thing that I have to. If I can drive one take-home message, we need to have our own data, our own studies, our own ethnic subgroups to say what is actually happening and see what is actually happening. It may be different. That's what I say. It may be different. It may be the same. But unless we do it in a wet lab, patients, real patients, we never know.
Sure, sir. So there is also another question from Dr. Sonal Bakshi. The question is, how are this kind of results treated by dynamic databases like FLINVAR? Genomic labs also classify results as per ACMG guidelines. Will it be nice to include such outlier findings with possible clinical significance?
That is very important. Uh, uh, the ACMG guidelines are very clear about what you have to classify as this tier 1, tier 2 type. That's what I understand from that. And just because we haven't done any studies, our mutations never appear in any studies. Unless we study our mutations and they publish our findings, our mutations will always remain as outliers. They are nowhere. So the onus of responsibility is upon us to identify our unique mutations and to publish them and possibly look at them in different angles. For example, now these mutations actually we can do in vitro studies. We can look at the type of biological behavior, looking at mice models. That is possible. Now, so basically, this unless we have our data will never come out. We will always have to deal with some data which may or may not be relevant to us.
Please go ahead. I see you have raised your hand. Tar, please go ahead.
Hi, good morning, all of you. I'm joining from New York, and I'm very sorry to miss the complete lecture I just joined. And I'm very much interested. The pole mutations, those are equally distributed in in Indian states like north to south. So, is there any similar association of the pole mutation from North India to South India? Because I also studied in my graduation in India, there are certain genes, they are differently associated with the metabolic diseases from south to north.
Very, very interesting. Thank you, Dr. Athar Singh. Actually, the only published study is one from CMC Vellore, Tamil Nadu, and they have got a cohort of 50 patients. And in the only other study is what we have done, to the best of our knowledge. We don't have any other published study from India. So this would be another question that will be very interesting to look at. So maybe all the Institute of Medical Sciences, PGI, or some good institutions can actually do this, replicate the study or different manner, NGS probably. And unless that data comes, we don't know actually. They might be having this pathogenic subtypes, and they could also be having good prognosis. So unless we actually have a good study from north, we will not be able. We will not be able to do the guesswork whether this, just like this, whatever is done in the West cannot be translated to our country. Whatever is happening in the South may not be translatable to North. So that is, but again, that is, we have to have our data. So as of now, we don't have data from North.
And another things, I just go through the pole gene and mutation on using the CBio portal. So I found that the maximum mutations are the missense. So at the end of the lecture, you mentioned that the most of the mutations are non-pathogenic. So my question is, these mutations can be a carrier mutations?
Yes, that is one question I have to answer. Whether these are your question is whether I tried to answer that question. Your question was is whether it is actually just an innocent mutation, just like non-drivership. These mutations that we have studied is actually within this 271 to 285, which is one of the most conserved segment in the exonucleus domain, that they are having significant function. And if at all there is any change in that mutation, in that sequence, there should be some biological effect. That is logically thinking. Number one. Then the study that came from CMC Vellore, they have a subgroup which are having POL and P53, which are having the worst prognosis. If the mutated, the poor prognosis was because of the P53 mutation, the graph should have followed the P53 graph, or it would be parallel the P53 mutated subgroup. It is not like that. They are having a different graph altogether, and that graph is much worse than P53. So we will definitely come up with our data in a short time. So we will know what actually our data says. Even the co-occurring mutations, whether they are actually driver mutation. Actually, the same question was posed by Cyriac Kandot, who is the first author of the Nature article. So I don't know the answer to that question actually. It is possible, but with the present scenario, I don't think that is this case. This is something unique. That is what my take on this at the present moment.
Thank you so much.
The next question that we have is, were the non-pathogenic mutations deemed benign in other population, or were they just variants of uncertain significance?
Many of the mutations that we have identified, except 10 of the mutations that we had were actually variants of unknown significance, VUS. And the rest of it were novel mutations. And they were recurrent. And these novel mutations were found to be pathogenic by more than 5. Actually, we used a cutoff of 3, more than 5 in silico tools. So almost all of them were branding this or giving a pathogenic variant outcome for these mutations. So because we do not have other studies from our ethnicity, we actually had to call these mutations, both VUS and those recurrent mutations, as significant. So the only term that we could use to identify these mutations are by what you call those non-pathogenic, because they were actually considered as non-pathogenic by the West. But what is significant is that whatever be the term by which we call them, these patients carry bad prognosis. They are different from the rest of the patients. And why they are different from the rest of the patients is something that has to be studied.
The next question that we have with us, are there any molecular classification system available for endometrial cancers?
Yes, molecular classifications. TCGA has actually classified into ultramutated, hypermutated, low copy number high. And also the PROMIS group, which is PROMIS classification by the Vancouver group, is actually more clinically suitable. That's what we actually did. In the sense, we will not be able to do NGS in all patients. So we actually did a limited sequencing using Sanger, and MMRD we identified by immunohistochemistry. And IH and P53 we actually did both immunohistochemistry and POL mutations. Yeah, but practically we won't be able to do both. We will have to do with only IHC 53. So we have the next generation sequencing methods as well as the PROMIS method for classifying them into TCGA subtypes. But for the POL mutated subtype, that the the current guideline that the West is now, especially the Leon Caslow and Jessica McAlpine, is actually suggesting that you should be doing the next generation sequencing to classify them correctly as pole mutated based on some pole scoring they have come up. So unless we have more studies, we won't be able to say what is the relevance of that finding. So basically, we have to now go by promise classification. That is what we have done. Or you can also do NGS if you can.
I hope this answers Priya's question. The next question is by Ami Singh. Can the presence of pole mutations affect decision making in endometrial cancer for treatment,
Oh, yes.
especially with chemotherapy?
Oh, yes, it is very significant. What disturbs us is the both ESGO-ESTRO guidelines and the FIGO new classification is clearly giving a hint to the clinician that pole mutated are having good prognosis. So the clinician has to discern that this pole mutations actually can be this pathogenic entity and a non-pathogenic entity. And those patients who are having non-pathogenic entity has to be treated in a very unique manner. We don't know what it is, but we have to go by standard treatment. At least we should not be treating them as good prognostic and don't treat them. They have to be treated just like at least the standard treatment they should get. So if you ask me whether this classification is important from treatment point of view, I think that is the most important thing. Because the patient should not be denied a treatment which is standard, because the ESGO-ESTRO even the ESMO actually has come up with guidelines whereby which they are de-escalating the treatment in stage 1 and 2 pole mutated patients, irrespective of their invasion to myometrium, LVSI, and other pathologic relevant things like non-endometrioid cancers. Whatever be the pathology, they say the pole mutated doesn't require treatment in stage 1 and 2, which is, I would say, it is very difficult to understand or practically apply. But in West, they are doing it. But our study at least is a very clear indicator that we cannot just copy or just follow the West in this aspect of treatment.
Yes. And do we have any targeted therapy for endometrial cancer?
Yes, especially in MMRD group, we have pembrolizumab, or what do you call this? Because they are actually because because of the high mutation burden, this WBCs are actually masked by a lot of new antigens. So this use of PD-L1 inhibitors actually help us in rejuvenating the innate immunity of the WBC. So that is also another thing that is also effective in pole mutated subgroup, not only in MMRD, in pole mutated subgroup. So we have to look at the effect of instead of using the conventional chemotherapy agents, where you can use targeted chemotherapy in these patients as well as MMRD group and achieve better outcome, especially in advanced stages. That will be a very interesting question to answer, especially in our patients.
Thank you, Doctor, for your invaluable time and the questions that you have answered. I think we have two more. We can take up two more. The next question is the mutations that we talked about before, were they constitutional or somatic in nature?
Somatic.
Somatic.
We haven't looked into the germline mutations. We have somatic.
Okay.
Tumor mutations.
And till when we can have our own data compared to can compare it to the Western data?
Yes, the answer is the answer is the onus of responsibility is actually upon each one of us. We have to do our own research, our own patients, and actually come up with our own publications. In the sense, we have to have our data. It's very, very important. Unless we have data, we will not go any step forward. We need to have our own data. More and more researchers have to do this genetic studies, and even they can actually do it, as you said, the targeted therapy can be tried in these patients. That will be very interesting and very useful to the patients also, except for the cost that is involved.
Let's take up the last question. The last question is, has any epidemiological studies been conducted in Kerala population for increased incidence of cancer?
I don't know. To my knowledge, no. Basically, what we think is that we have very high incidence of lifestyle diseases in Kerala. The obesity rate is around 38-39%, and the diabetes is around 29-27%. It is actually bordering onto West, a lot of Western countries, and that it's the obesity epidemic actually with the cardiovascular disease, just like endometrial cancer. Endometrial cancer was a rare cancer around 15 years back. Now I operate around 4 or 5 patients per week. So endometrial cancer is not very rare nowadays. It is coming up, increasing with increasing obesity trends and hypertension, diabetes, lifestyle diseases. So epidemiological study will be a very, I think, a need of the hour. We need to look at it from that standpoint. Across the population, we have to look at it. We still, to my knowledge, do not have a population-based cancer data in the sense. So from RCC, Malabar Cancer Center, and Regional Cancer Center, we have hospital-based data, but population-based data on cancer registries are not, practically it is not there. So that will be very interesting to have such data, because we will be able to see the cancer in its real context. Underlying context may be the lifestyle. For example, especially the endometrial cancer, it is related to the lifestyle, the obesity and hypertension, diabetes. So, so that will be putting, that study will be putting the cancer in its, the increasing cancer trend in its proper perspective.
Thank you, Dr. Santosh, for your time and enlightening us on the prognostic significance of polling mutations.
Thank you.
We know we need more data in Indian population, and we need to do more broad testing using the NGS approaches to at least take care of this gap. Also, I would like to extend my sincere gratitude to the attendees for their active participation. And I also encourage each of you to share your questions if left unanswered on the Karkinos YouTube channel and provide feedback to help us in continuous improvement. I trust that the session has contributed to knowledge, and I look forward to everyone's engagement in the upcoming webinar. Our next webinar featuring Dr. Saurabh discussing CAR T-cell therapies is scheduled for May 11th. We hope to see you there. With that, we conclude today's session, and thank you all for your participation, and thank you, Dr. Santosh, again.
Thank you.
Thank you, everyone.